Good morning, everyone. Thanks for your kind introduction, and thanks for this opportunity to speak here at the conference. Today, I would like to tell you about my postdoctoral work I've been working on the past few years, basically looking at genomic applications and merging them with CMOS technology. I would like to take you through a journey, and it's not going to be a linear journey, just to see how the technology development evolved. First, I need to tell you a little bit about DNA sequencing technique, which I utilized this platform before. I looked at Cas9, another genome editing application. So DNA sequencing is very important since it contains the heritable genetic information and it contains the genetic blueprint who we are. It is composed of 4 basic building units, A, C, T, and G, and it forms the DNA molecules, and physically they form a double helix. Now, it's been shown by medical professionals that mutations in these genes can be linked to diseases. And understanding the full genomic scope and mapping the full genome is very important to understand the next phase of medicine, which is called personalized medicine, where we can sequence everyone's genome for a very low cost, identify these mutations, and hopefully come up with predictive models to correct for these, or even come up with tailored treatments. So genome sequencing is very important, and I just would like to show a complementary method. Genome editing will be also very important in the next century. So clearly, enabling technologies are needed to realize this goal and to address many major challenges associated to disease. I just want to give you some historical trends in DNA sequencing cost. As you can see, when the $1,000 genome project was introduced, the genome sequencing cost drastically decreased. With technology advancement and the introduction of Illumina sequencing in further decreased, and when CMOS technology or integrated circuit technology was introduced, currently I'm showing an example here from Oxford Nanopore, you can see that the cost drastically reduces. So in order to realize DNA sequencing and DNA analysis in general, we need to have these enabling technologies and we need to reduce cost. So when we look at the historical improvement of DNA sequencing speed, it follows an exponential increase. Similarly, when the 3rd generation sequencing devices, these nanosensors, CMOS integrated circuit sensors, are introduced, you can see that the speed magnificently increases. So what I'm trying to point out here with technology development and CMOS sensors, integrated sensor technology, which is currently being developed in cell phone and computational chip technology, will enable also genomic analysis in a more affordable way. Just giving you a survey here on current 3rd generation DNA sequencing techniques. And what I'm trying to point your attention to is that a physical sensor in an array setting is typically coupled to an enzyme which performs some kind of functions. For example, sequence DNA in this particular example. But you can imagine that could be any other enzyme which actually processes DNA. And there are many of them. One of them is also Cas9. And I will show you in later slides how I'm envisioning this technique. So the molecular details for sequencing of a DNA polymerase, which requires a template, and it can synthesize a new template complementary to this strand. So it replicates DNA. Okay? This is Illumina sequencing, what I'm showing here. You need to immobilize single-stranded DNA on a substrate. You have a special DNA molecule which has a fluorescent modification on it and a blocking group. When you introduce the DNA polymerase on this chip and the modified nucleotides, at the first stage, the complementary nucleotides will incorporate. Using fluorescent excitation, we will detect at what particular location or DNA strand the identity of the nucleotide is. Then we can remove the nucleotide blocking group and also the fluorophore to finish our first cycle, and we can repeat the second, the third, and the fourth base identification the same way. So this is how the current Illumina sequencers work. This is dominates probably 80% of the market currently. Just to understand it a little bit better visually, so we have DNA molecules here currently invisible, but that's how we're identifying the bases one by one, and we're using image processing to create these large maps and assemble the DNA molecules. This is, for example, an example of the Illumina chip under a microscope. Nanopore sequencing has great potential, so I would like to shift a little bit after the introduction of sequencing. And essentially, the idea here was to let's form a lipid bilayer, let's set up a voltage gradient between the two chambers, cis and trans side. Let's use electrolyte solution, and let's have a single-stranded DNA translocate through. Why it will translocate through? DNA is negatively charged. The bases will be identified as current blockade signatures or disturbed, while the DNA bases move across the pore. That's the basic principle. Very exciting opportunity, but very challenging too. Exciting because it's a label-free technology. Challenging because you need to interpret the measure signal to a molecular property. So it's not straightforward. A lot of advantages associated with it. No amplification, extended read length. And these are just some of the examples what current nanopore-based detection are based on. I'm going to focus on a biological nanopore, and I'm going to give you a brief description what we already published before. And it's going to be a nice bridge to the Cas9 work I'm briefly will describe. So we connected a single DNA polymerase to the protein pore. We embedded this in a lipid bilayer over a CMOS sensor array. So below every protein construct, there is a sensor, and I will detail it a little bit more in the next slide. We have 4 tag nucleotides. When they incorporate, the tag will be captured in the pore lumen, which will give rise to a current blockade signature. When the catalytic cycle ends, this tag will be removed. That means that the signal will be bouncing back to open channel reading. So essentially, these responses will determine, and the different type of tags we selected can give 4 different current blockade signatures. This is the basis of the technique we developed. Obviously, very important here to come up with a conjugation strategy that you have only one polymerase on these little nanopore sensors. My postdoc colleague Ben Strain just carried this out. And these are the structures just giving you an idea for the tags. So this is the nucleotide itself, and that's how we're connecting the tag, which is about 30 base pairs long. When you're looking at the electrostatic distribution along the line, you can see that it can be varied. So we can design these, and they will interact differently when they're captured in the pore lumen, giving rise to different current blockade signatures. This is the measurement setup that I mentioned before. We have an alpha hemolysin. We have a connected polymerase. We have a lipid bilayer. We have a very small thin film here. This is the cis chamber. Voltage source. Okay. And this is how the circuit looks like. So this is a patch clamp amplifier. So the signal will be amplified and compared to a set value. And essentially what we're gaining here is picoamp scale responses. Okay? As something, a polymerase blocking the electrolyte flow. So microfabrication will be carried out with basic photolithography. I'm just zooming on one of these single cells. That's how they look like. This is the working device, just giving you like a physical understanding that this is the DNA sequencer now. And this is what the CMOS technology enables. And when we have, oh, really? So when we have, isn't that 15 minutes? Sorry. Okay. So when we have the DNA polymerase connected to the pore, then we can see that we have these distinct current blockade signatures. So let me go to the Cas9 description. So very similarly, sorry, I need to. Sorry. Okay. So very similarly, we hooked up a Cas9 molecule to the pore. Okay, but we're using a different circuitry here. We actually applying a voltage across the lipid bilayer and the pore. We open the circuit, and essentially it's going to decharge, and we're going to monitor the voltage decay. What will be associated to the resistance of the nanopore, or whatever state the nanopore is in. So is something captured there or not? So this is the waveform we're applying. So it has a bright cycle and a dark phase. 200 millivolts switches between 200 and minus 10 millivolts for 8 milliseconds and 12 milliseconds. So this way, we will be able to actually capture the tag nucleotide multiple times, not just once, as in the DC capture case. So what you can see here is that this tag is being captured multiple times. Looking at these capture profiles, we can define kinetic properties. These are kind of arbitrary. Okay, let me skip through the Cas9 work. And very similarly here, what we did, so very similarly what we did, we have a Cas9 molecule which can be programmed to target a particular genomic region using a guide RNA. So this is the nomenclature that I'm going to use. The experimental design is very similar to the nanopore sequencer I described. My colleague helped me to conjugate a Cas9 here now. These are the basic components, the guide RNA, the target RNA. So the principle here of the detection is also based on tags. So we put 3' extended tags on the guide RNA and also on the target RNA. And the principle of detection looks like this. So when it's on the guide RNA, we're going to perform this alternating wave function, which will capture the tag, release the tag, capture the tag, release the tag. It can give you the information about the guide RNA presence. When you have the 3' extension on the target DNA, it will give you information about if that particular genomic locus is present. And at the end of the catalytic cycle, actually, it will be cleaved off, and you can monitor the cleaving efficiency. So what I showed here theoretically is single molecular guide detection, target DNA detection, and also cleavage modification. So these are the preliminary results I have. So what we've shown here, that when we have a guide RNA with a tag, we can detect this very nicely on a single molecular level. When we have a polynucleotide tail on the guide RNA, we can also detect this. And obviously in control experiments where there's no tag, they cannot be detected. And if you can imagine, in future research, can be carried out where you actually design these Cas9 variants using this technique on a large CMOS array. where you can introduce specific mutations and you can monitor their enzymatic activity.